Hey future doctors, today we are going to see about bacterial classification. So this is the most important table. First is two kingdom classification was given by Linnaeus and it was divided into Vegetabilia and Animalia. Three kingdom classification was given by Haeckel, which is divided into Protista, Plantae and Animalia. Then two empire given by Chaton, which is divided into Prokaryota and Eukaryota. Then four kingdom classification was given by Copeland, which is divided into Monera, Protista, Plantae, Animalia. Then five kingdom was given by Whitaker, divided into Monera, Protista, Plantae, Animalia and Fungi. Then three domain was given by Woos, which is divided into bacteria, archaea and eukarya. Then sixth kingdom was given by Cavalier and Smith. It is divided into bacteria, protista, chromista, plantae, fungi and animalia. So this was all about various kingdoms and scientists. Let's discuss about Cavalier and Smith classification. It was a six kingdom classification divided into bacteria, protista, chromista, plantae, fungi and animalia. The hierarchy, it is in the decreasing order, phylum, division, class, order, suborder, family, tribe, genus and then species. So now let's discuss about morphology of bacteria. Depending on the shape, bacteria are classified as cocci, bacilli or rods. So cocci are the bacteria which is oval or spherical in shape and bacilli or the rod are rod shaped. Then cocci can be arranged in groups as a clusters, pairs or chains. As you can see, bacilli also can be arranged in chains, pairs and some bacilli are also curved, comma shaped, cuneiform shaped. So as you can see in this table, this is clusters, this is chains, this is tetrad, octet and pairs. Same as this is in pairs, this is in bacilli in chains and this is cuneiform patterns. Then these are bacilli, this is a comma shaped and this is spirally coiled. So these are different type of cocci and bacilli which are arranged in different types. Then depending on the gram staining, it is divided into gram positive cocci, gram negative cocci, gram positive bacilli and gram negative bacilli. Now there are some exceptions in the gram staining. So some bacteria are usually weak gram stained and hence it requires special stains for their demonstration. Example spirochetes. Example is Treponema and Leptospira. These are very thin spirally coiled bacilli, so it requires special stains. The next is Mycoplasma. Here the cell wall is deficient, so it also requires special stain. Is Rickettsia and Chlamydia. These also require special stain. So basically there are three examples which are weakly gram stained. Now let's see about the bacterial cell anatomy. So outer layer or the envelope of the cell, it consists of the rigid cell wall and then plasma membrane. Then the cytoplasm, it consists of cytoplasmic inclusions, mesosome, ribosome, granules and vacuoles. As well as it also contains diffuse nucleoid containing a single circular chromosome. So as you can see here, these are ribosomes, this is a nucleoid, then these are inclusion bodies and this is a capsule which covers the bacterial cell, then this is a flagellum and this is a plasma membrane as well as this is cell wall. So some bacteria also possesses additional cell appendages such as capsule, flagella and fimbriae as cell wall. So it is a tough rigid structure surrounding bacterium. There are so many functions of cell wall, so it provides protection to the cell against osmotic lysis. It also confers the rigidity. It also protects from the toxic substances. As well as cell wall also contains the virulence factor endotoxin. Then cell wall, it can be divided into gram positive and gram negative. So in gram positive bacteria, it is simpler than that of the gram negative bacteria. It is made up of peptidoglycan. In this, the layer is much thicker than the gram negative cell wall and each layer, it consists of mucopeptide chain which is composed of N-acetylmuramic acid NAM and N-acetylglucosamine NAG molecule which are cross-linked to each other via tetrapeptide side chain and pentaglycine bridges. This figure, as you can see, this is a peptidoglycan layer of gram positive cell wall. So these are NAG, this is NAM and these two molecules are cross-linked by the tetrapeptide bond as well as this is a pentaglycine bridge. Next is tetrapeptide chain. It consists of L-alanine, D-glutamine, L-lysine and D-alanine. So again you can see in this figure that this is L-alanine, this is D-glutamine, this is L-lysine and this is D-alanine. So next point is L-lysine of the one tetrapeptide chain is covalently linked to terminal D-alanine of the adjacent chain via pentaglycine bridges. As you can see that this is L-lysine and this is D-alanine, it is connected by the pentaglycine bridges. One more acid which is present in the gram positive cell wall. So it is only present in the gram positive cell wall, it is absent in the gram negative cell wall, that is teichoic acid. It is a polymer of glycerol or ribitol which is joined by the phosphate groups. So as you can see, this is a structure of gram positive cell wall. This is a teichoic acid. And this may maintain the structure of the cell wall. There are two types of teichoic acid. First is lipoteichoic acid and next is teichoic acid. So this was all about gram positive cell wall. Now we are going to see about gram negative cell wall. It is much thinner and more complex than the positive cell wall. It also comprises of the peptidoglycan layer which is very thin and it is composed of mucopeptide chain which is similar to the gram positive cell wall and consists of alternate NAM and NAG molecules. It differs from the gram positive cell wall as it has mesodiaminopimelic acid which is present at the third position of the tetrapeptide side chain which is ascended from the NAM molecule. So here as you can see, this is L-alanine, this is D-glutamine, this is mesodiaminopimelic acid and this is D-alanine. Additional to this, the pentaglycine bridges that is also absent. So as we have seen in the gram positive cell wall it was present, but in gram negative it is absent. Then the tetrapeptide side chain are directly linked to each other by covalent linkage between D-alanine of one chain with the mesodiaminopimelic acid of the adjacent chain. Here, as you can see that the D-alanine of the one chain with the other chain of the mesodiaminopimelic acid, they are connected by the tetrapeptide side chain. It was all about cell wall of the gram-positive and gram-negative bacteria. Now next topic is outer membrane. So phospholipid layer which lies outside the thin peptidoglycan layer, it is firmly attached to the latter by the membrane protein and it is known as bronze lipoprotein. The outer membrane also serves as a protective barrier to the cell as well as the outer membrane protein or porine proteins. These are the specialized proteins which are present in the outer membrane which help in transport of the smaller molecules. Then next is lipopolysaccharides, LPS. So this layer is unique to the gram-negative bacteria and it is absent in gram-positive. Of three parts, lipid A, core polysaccharide and O side chain. So lipid A has the endotoxic activities such as tissue necrosis and anti-tumor activity. Then core polysaccharide, it is projected from the lipid A region and it is composed of 10 to 12 sugar moieties. Then O side chain, it is a polysaccharide side chain which extend outward from the core polysaccharide region. So O side chain also varies in the composition between the bacterial strains. Now next is periplasmic space. It is a space present between the inner cell membrane and outer cell membrane. It also encompasses the peptidoglycan layer. Differentiating features between the gram-positive and gram-negative cell wall. So peptidoglycan layer, in gram-positive cell wall it is thicker and in gram-negative cell wall it is thinner. Then at the third position of the tetrapeptide chain in gram-positive cell wall, L-lysine is present, but in gram-negative it is mesodiaminopimelic acid. Then pentaglycine bridges, they are present in gram-positive, but it is absent in gram-negative cell wall. Then lipid content. In gram-positive cell wall, it is nil or scanty, just 2 to 5%, but in gram-negative cell wall, it is present and it is 15 to 20%. Next is lipopolysaccharide. It is absent in gram-positive cell wall and it is present in the gram-negative cell wall. Then teichoic acid, it is just present in gram-positive, absent in gram-negative. Then variety of amino acid, there are very few in gram-positive and there are several in gram-negative cell wall. The last one is aromatic amino acid. It is absent in gram-positive cell wall and it is present in gram-negative cell wall. So this was all about cell wall. Now next topic is cell membrane. So as we know that the fluid mosaic model, it is most widely accepted model about the cell membrane which describes the cell membrane structure. It composed of bilayered phospholipid in which several proteins are embedded, integral as well as peripheral proteins. So here as you can see this is an integral protein and these are peripheral proteins. It differs from the eukaryotic membrane in lacking sterols such as cholesterol except in mycoplasma. However, many bacterial membrane do contain pentacyclic sterol-like molecule known as hopenoids. There are some functions of the cell membrane. First is it is semi-permeable membrane which acts as an osmotic barrier and it allows selectively particular ions or molecules to pass. The next is transport system. The proteins and enzymes present in the cell membrane, they are involved in nutrient uptake and waste excretion. Additional to this, cell membrane also has one function that is the site for the metabolic processes such as respiration, synthesis of lipid, cell wall, probably chromosome segregation. So this was all about part 1. In next part we are going to see about cell wall appendages.